Hi, hello, my dear students. This is your Prasanna Harikrishna sir, Assistant Professor of Botany, SRR Government Arts and Science College, Karimnagar. So today we are going to see cyclic photophosphorylation. So already we have gone through non-cyclic photophosphorylation in my previous session. Clear? So today we are going to see the cyclic photophosphorylation. So as you know, phosphorylation nothing but what? Formation or production of ATP. Then production of ATP in the presence of what? Light. Okay, so presence of production of ATP in the presence of sunlight is called as what? Photophosphorylation. But here cyclic photophosphorylation. Okay, so photophosphorylation takes place in cyclic manner. Cyclic manner means what? For example, this is substance A. It loses electrons. These electrons move through different complexes and finally the electron reaches to complex A only. Okay, so wherever the electron removal or loss takes place, this electron can be substituted through the different moments or different mechanisms. So this electron finally reaches to same complex, that is complex A. So hence it is cyclic manner. So this kind of phosphorylation can be called as cyclic photophosphorylation. So then come to the contribution of scientists, that is Frankel. Okay, so Frankel had done a very good research over cyclic photophosphorylation on chlorella, and there is another bacteria that is more important than chlorella, that is Rhodospirillum. What? Rhodospirillum rubrum. Rhodospirillum rubrum, so is a kind of purple non-sulfur bacteria. So this is what? So purple non-sulfur bacteria, that is Rhodospirillum rubrum. So on which Frankel had done a great experiments of cyclic photophosphorylation. So he concluded that the electrons move from one complex, then these electrons gone through different systems, finally they reach to the same complex in a cyclic manner. So how it looks like? Remember, so in this particular cyclic photophosphorylation, only one photosystem is involved, that is PS1 only, right? The reaction center of PS1 should be P700. Okay, P700. For example, if you see the diagrammatic representation, these are all the chlorophyll molecules, right? This is chlorophyll A, means reaction center. Okay, this is chlorophyll A, that is reaction center. What is the reaction center? P700. Okay, so whenever it receives sunlight, that is H nu. So whenever this particular complex, that is photosystem 1, the reaction center will be P700. This is completely PS1. So photosystem 1, so in which the reaction center is P700. Whenever it receives sunlight, so these are all the electrons. This sunlight moves from one chlorophyll molecule to another. Finally it reaches to chlorophyll A molecule. So when it receives an energy, the outer orbital is there now. So electrons are there in the outer orbital. So whenever it receives energy, it will be in excited condition. Okay, it becomes excited. Then the electrons are lost. Right? Electrons are lost. These electrons are received by iron sulfur protein. So this is what? The first electron receiver in PS1, or you can say cyclic photophosphorylation. Okay, so in cyclic photophosphorylation with respect to PS1, the first electron receiver will be iron sulfur protein. Okay, so electrons are lost whenever the chlorophyll A molecule becomes excited in condition. Okay, now these electrons move to the next electron receiver, that is ferredoxin. Then followed by cytochrome B6. Cytochrome B6, site B6. Okay. So whenever there is movement of electrons from one complex to another, particularly ferredoxin to cytochrome B6, there is a formation of ATP from ADP. Okay, so one ATP molecule is formed from the ADP and phosphate. Then this electron is moved to cytochrome F. Cytochrome F. So whenever there is a movement of electrons from cytochrome B6 to cytochrome F, there is again a formation of ATP from adenosine diphosphate and phosphate. Okay. Then cytochrome F2, PC. So PC stands for plastocyanin. So this is copper containing complex. Then plastocyanin 2, finally that is PS1. Electron finally reaches to the same complex, that is photosystem 1. So this is all about cyclic photophosphorylation. Simple. So two ATP molecules are formed in this particular context. Clear? There is no, remember, only ATPs are formed in the cyclic photophosphorylation. Clear? Only one system is involved, that is PS1 is a only involved system. There is no involvement of PS2. If there is no involvement of PS2, there is no OEC, that is oxygen evolving complex, means there is no creation or formation of oxygen. Okay, so oxygen cannot produced in this particular cyclic photophosphorylation, whereas non-cyclic photophosphorylation, oxygen is created from PS2, that is photosystem 2. Okay, and there is no production of NADPH2 in this regard. Okay, only ATPs are formed. There are no formation of NADPH2s, and there is no formation or creation of oxygen, and only one system is involved, whereas in non-cyclic photophosphorylation, two systems are involved. PS1 and PS2 both are involved in non-cyclic. Only PS1 is involved in this cyclic photophosphorylation. Clear? So this is all about cyclic photophosphorylation. So after completion of cyclic photophosphorylation, finally we have we have finished the first phase of photosynthetic process, that is light phase. So in the next session we'll go to dark phase. So dark phase of photosynthesis, we will have C3 cycle, C4 cycle, C2 cycle and CAM cycle. So different cycles we will be discussing in the further classes. So thank you one and all.